populate, and then I'll start introducing folks. All right, while the rest of people stream on in, I'll get started here. Hi, everybody. Good morning, good afternoon, or good evening, wherever you may be. Welcome to the 12th week of the fall 2020 iteration of the Long-Term Animal Research Seminar Series. This week, we're going to try something a little bit different. We're going to have 4 different talks that describe research done in the same study system, the Mara Hyena project. In this case, people will be asking questions in the same system from different angles. So we'll hear a brief introduction to the system first from Dr. Holekamp, followed by 15-minute talks from Dr. Tracy Montgomery, Connie Rojas, and Dr. Strauss, and then we'll finally return to Kay for a longer talk at the end. If you have strong feelings about this new format, please let us know either via Twitter or email, because it could serve as a model for subsequent talks and approaches going forward in this series. And related to that topic of feedback, we're currently soliciting feedback about the seminar series via a brief survey. You can find that survey pinned at the top of our Twitter page. It's also in the description of the YouTube video for this week. And I'm going to post it in the chat right now, so no one can say that they didn't see it. So it would really help us if you all could take 3 to 5 minutes sometime in the next couple of weeks to fill out that survey. It's really important for us both to assess our success at achieving broader impacts goals, as well as for charting a path forward beyond this semester. If you're participating via Zoom, you should see a Q&A tab on the top or bottom of your screen. If you open that tab, you'll be able to type any questions that you have, as well as see and upvote other people's questions. We're going to have brief question and answer sessions at the end of each of the shorter talks. And then at the end of Kay's talk, we'll have a longer Q&A session where you can ask questions both of Kay, as well as particular questions of previous speakers if you didn't have time earlier. At that time, just type in the speaker's name before you ask your question, so I know who to address it to. And finally, we're going to post a recording of today's talk on YouTube. So if you know any who can't attend live, or if you want to rewatch this talk, that you can do so online as many times as you'd like. Okay, so now I'll get started and I'll introduce our first speaker today, who is going to be Dr. Holekamp, who's going to give us a brief introduction of the system first. Holekamp is a university distinguished professor in the Department of Integrative Biology at Michigan State University. Among her many honors, she's a fellow of the American Academy of Arts and Sciences, and last year at the ABS meeting, she received the Distinguished Animal Behaviorist Award, which is the highest honor in the Animal Behavior Society. For more than 30 years, Kay has led a long-term longitudinal study of hyenas in Masai Mara, Kenya. And over that time, she served as an example of extraordinary commitment to high-quality field research, and her work in the wild hyena system has made fundamental contributions to our understanding of the ecology and evolution of mammalian behavior, sociality, and life history. As evidenced by the other great talks that we're in store for today, Kay also has a reputation as being an extraordinary, generous, and committed mentor to her trainees, a devotion that was recognized when she was awarded Michigan State's inaugural Outstanding Faculty Mentor Award. So it is now with great pleasure that I welcome Kay today, and I'll turn things over to her now.
Thanks very much, Matthew. Now I've got to share, oh crap, okay, never mind. All right, we're just gonna go ahead without that. Sorry, anyway, thanks very much for inviting us. I think we're all very flattered and pleased to be here. And as Matthew said, what I'm going to do first is simply introduce the study system, and then everyone gets to talk. Spotted hyenas live actually in large stable social groups called clans that can contain up to 130 individuals, and each individual member of a clan knows all the other members individually, and together they defend a common territory. And they also rear their cubs together at a communal den. And one of the most interesting things about hyena society to me is that it doesn't look anything like the societies of any other mammalian carnivore that you can think of. These animals all live in relatively small groups, and at least within members of one sex, typically they are living with their close kin. Instead, spotted hyenas look in their societies like the societies of baboons and macaques and other Cercopithecine primates. And it's been rather extraordinary convergence, I think, in terms of group size and structure. We see in both baboons and spotted hyenas that mean within group relatedness is very low. We have multiple adults and multiple generations of both sexes. We see, as in most mammals, male dispersal and female philopatry. And most importantly, the societies of both these creatures are structured by hierarchical rank relationships. One thing that's very different from baboon societies is that spotted hyenas live in fission-fusion societies. And here I'm showing you an area of roughly 100 square kilometers, and we've got 6 adult females radio collared with GPS transmitters that give a location once a second. And you're looking at a speeded up version of the entire month of January 2017. We've got 2 high-ranking, 2 medium, and 2 low-ranking females here, and you can actually see that they're all coming together frequently over here, which is where the den is at this moment, but they go up alone to forage and stay out of conflict with their peers, particularly the lower ranking females. So we've been working in the Masai Mara National Reserve in Kenya since 1988. And these days we're working with 6 clans that contain somewhere between 4 and 130 individuals. The 6 clans you can actually see down here. And this is an area 2 degrees south of the equator. So we get abundant sunlight, and that produces abundant grass, which supports abundant herbivores, and those in turn support abundant carnivores. And we know each individual in each of our study clans by its unique pattern of spots and ear damage, so you don't have to mark them, they come that way. And we spend several hours every day, mainly around dawn and dusk, sitting in cars like this one, to which the hyenas have been habituated since they were babies. For the most part, they've just grown up with these cars and they just go about their business in front of us. But one thing we've done religiously since the start of the study is to record every single interaction of this type that's occurred in our presence. And what you're seeing here is a low-level fight between Cochise on the left and Oliver Twist on the right. And Cochise is exhibiting all the classic signs of hyena aggression. Her ears are cocked forward. She's lunging up out of this hole at Oliver Twist, who's interested in Cochise's baby. And by contrast, Oliver Twist has her ears plastered back. She's squinting to protect her eyes, tail down between her legs, and her whole body's crouched down and bent around towards Cochise in a classic hyena appeasement posture. And it's because Oliver Twist is appeasing Cochise, we call Oliver Twist the loser in this particular fight, the winner. Cochise is the winner. So what we've done here is to simply take all those fights, which is what these numbers in this matrix represent, where there was a clear winner and a clear loser, and listed the animals in the clan, although I've taken out about 50 hyenas here between Bailey and Midget, so you can see what's going on more clearly. But we've listed those as animals, as winners in these fights, from the animal who can win against everybody else in the society down to Boston here, who cannot win against anyone else in the society. And the same animals are listed as losers up here. So what you can see right off the bat is that this is a very clear linear hierarchy. There are very few fight outcomes that are inconsistent with the rank orderings that we've assigned here. And again, this is much like what you'd find in a macaque group, for example, except that in spotted hyena society, it's the breeding females who are at the top and the breeding males who are at the bottom. Most of the breeding males come in from elsewhere. So we see females being socially dominant to males. And I originally went out there in early 1988 to try and figure out how cubs slot into this system. Got stuck there, still there, but I just want to show you how it is that on average cubs do slot into the hierarchy. The hyenas exhibit a primate-like system of maternal rank inheritance, where if the top 3 ranked females in the clan, for example, are these 3, Murphy, Seinfeld, and Whoopi, and Murphy has a baby. She gives birth to Athena, and then Athena slots right, and Murphy takes Athena's side against Seinfeld and Whoopi, and Athena slots right in below Murphy, and she can dominate any lower-ranking female. But then comes the rub, where Murphy has another litter. She gives birth to Artemis, and now she takes Artemis aside even against Athena. So we see maternal rank inheritance with youngest ascendancy in the species. And because cubs slot in below their mothers, we see that all the members of any given matriline occupy adjacent rank positions in this hierarchy. So here, for example, is the members of the alpha matriline, which, because they have priority of access to food, is the biggest, and then that beta matriline, and so forth until you get down until the immigrant males at the very bottom of the society. In hyena life history, females typically give birth in an isolated natal den. They then transfer their cub about 3 or 4 weeks later to the clan's communal den. And I'm showing you a typical communal den scene down here. This is a place where everybody in the clan comes to visit periodically to meet the new cubs and to introduce themselves and stay socially connected. Then, after 9 or 10 months at the communal den, cubs start traveling around the home range, originally with their moms, later on their own. On average, they're weaned at 13 and a half months. They are both sexes are physiologically competent to breed at 24 months. And then females are philopatric, so they spend their whole lives in the natal territory. By contrast, almost 100% of surviving males disperse after puberty. And if they're lucky, they become integrated into a neighboring clan where they may reproduce. So these animals are very effective hunters. They catch 95% of their own food in our study area. But once an animal is brought down, then hyenas converge on the scene from everywhere, and there's a lot of fighting over the food, and outcomes of this competition over food are determined by social rank, because high-ranking individuals can use aggression to displace lower-ranking individuals, and that has profound effects on virtually every aspect of the animal's biology. So for example, I'm introducing you to Barbara on the right and Batman on the left. Barbara's the daughter of the alpha female in our clan at the time, and her mom can waltz into a kill made by anybody in the society and steal it from them and turn that into milk for Barbara, and Barbara effectively has access to ad lib food. By contrast, Batman here, her mom's 17th ranked in the clan, and so she's much lower feeding priority, and she grows much more slowly because these two girls were born 3 days apart, and this is what they looked like when they were 6 months old. So there's really dramatic variation in growth rates, and that affects age of first reproduction. So here I'm showing you the cohort in which we have Barbara and Batman, and this is looking at the age at which they first bear young. So look at this, here's Barbara. Oh, by the way, I'm using a primate-like convention of calling high ranks 1 over here, and low ranks these higher numbers. In some cases you'll see relative ranks shown, and that's going to have the opposite pattern. But in any case, this is Barbara here. So you can see that before she's 2.5 years old, she gives birth to her first litter. By contrast, here is Batman. At 72 months of age, she first has her babies. And those differential, that affects the length of the reproductive lifespan. And not only are high-ranking females breeding for more years, but they're actually producing more young per year, as you can see in the right panel here. So this has profound long-term fitness consequences, and I just want to show you what I mean by that. Lawrence Frank worked with our original study clan, starting with his PhD work in 1979. And when he went out there, he found 19 females, these 19 females. And now what we're looking at is at decade-long intervals, we're looking at the representation among the adult females present in the clan of the descendants of those original 19 females. And so you can see that by 40 years later, there are only descendants of 4 of those original 19 females represented in the clan at all, and K-Butt's descendants represent over 60% of the adult females present in the clan. So these gray triangles then represent extinctions of entire matrilines over time. And with that, I will stop and turn the stage over to Tracy.
Great, thank you, Kay. We'll come back to you once we hear from the others. It's a great introduction. Okay, so our next speaker is Dr. Tracy Montgomery, who received her PhD from Michigan State earlier this year. She's now a postdoctoral researcher at the Max Planck Institute of Animal Behavior. Tracy is broadly interested in the evolution of sociality and the forces that operate across time and biological organization to produce complex societies, including some really interesting current work studying the predator-prey dynamics of chipmunks and fossa. It's great to have her here, and I'll turn things over to her now.
Thanks, Matthew. Hi, I'm really excited to be here today telling you about my work investigating collective action in complex societies. Before I start, I'd like to acknowledge Kenna Lehman, who's been a collaborator on this project from the beginning. Group coordination and cooperation are interesting because they require that multiple individuals coordinate their behavior to produce an outcome that could not be achieved by a single individual acting alone. Across species, individuals and social groups cooperate when acquiring resources, when defending themselves and their offspring, or when engaging in territorial defense. Even in species in which these cooperative behaviors appear to be collective, individual participation is often highly variable, and it is rare that all group members are active. Cooperation is thus usually not a true collective action involving all members of a social group, but is rather joint action by a subset of individuals. This brings us to a paradox known as the collective action problem. Why in the world would an individual choose to be this meerkat in striking range of the cobra, rather than this meerkat hanging out at the back, For this type of collective behavior to evolve, individuals must experience divergent costs and benefits, such that it is worth it for the close meerkat to take that risk. So today, we're going to investigate collective action and its potential divergent benefits using a long-term data set from wild spotted hyenas living in the Masai Mara National Reserve. Despite low mean relatedness among clanmates, spotted hyenas frequently cooperate in coalitionary aggression, which maintains the dominance hierarchy, in the acquisition and defense of food, in territory advertisement and defense during border patrols and interclan wars, and when interacting with lions. Lions and spotted hyenas are the largest and most abundant large carnivores in the East African ecosystem. The two species are competitors. They have a dietary overlap of 70%, and both species kleptoparasitize and scavenge from one another. Lions are actually the leading cause of mortality in many hyena populations, and an attack from a lion can result in serious injury or death for a hyena. Despite these risks, hyenas often interact with lions and frequently engage in cooperative mobbing behavior, which occurs when two or more individuals in a high state of arousal synchronously approach or attack a threatening stimulus in the environment. So now I'm going to show you a slideshow depicting a high-intensity lion-hyena interaction. Here, a photographer has documented lions killing a kudu in Etosha National Park in Namibia. Our first hyena arrives and begins to whoop to call in other hyenas. Within 10 minutes, 16 hyenas were present, and the lions charged at the hyenas. The hyenas charge back, and the lions defend. There are clear dangers to individual hyenas when mobbing. A bold hyena even tries to steal a bite, but it comes at a cost. Luckily, that hyena had group mates to back it up. Now the hyenas have gained control of the carcass, and they're quickly demolishing it. So hopefully you now have a better idea of what some lion-hyena interactions are like. They're crazy. And we can move forward to address two questions about collective action during lion-hyena encounters. First, what traits affect participation in cooperative mobbing behavior? Second, who benefits from cooperative mobbing? Starting with question one, we can simply ask, who participates in mobbing? We use a data set of all lion-hyena interaction sessions in which at least one mob occurred. The data set contains all mobbing events at these sessions, as well as all hyenas present during each mobbing event. Because mobbing is a relatively rare behavior, our data span 29 years in 4 clans, and we couldn't have done this study without this long-term data. Our final data set consists of close to 5,000 records or focal individuals, where for each of 344 mobbing events, we have a list of mobbing individuals or cooperators, and a list of individuals who were present but did not mob, or defectors. Individuals appear in the data set multiple times, since the defining feature of the data set is each mob. Using this data set, we address 3 non-mutually exclusive hypotheses about what individual attributes and social contexts lead to participation in cooperative mobbing behavior. First, we hypothesize that individual socio-demographic traits may affect decisions to participate in mobbing. We measured 3 attributes: sex, age, and social rank. Our second hypothesis is that individuals who engage in real-time affiliative interactions may be more likely to participate in cooperative mobbing. Here, we quantified affiliative behavior by coding whether a focal individual was involved in a greeting ceremony prior to a mob. Our third hypothesis suggests that individuals who are close social allies or kin may be more likely to mob together. We measure social bond strength using the association index, here calculated as the average association index of the focal individual with mobbers in the year prior to the mob. Our second measure of long-term social relationships is that of kinship, or the relatedness of the mobbers. Here, we define relatedness as the proportion of mobbers who are maternally related to the focal individual. In our analysis, we performed a series of logistic mixed effect models where we modeled the probability of an individual mobbing as a function of these variables. For each model, we created a global model and then used AIC criteria to select the top model, including session, mob, and hyena as random effects. As a quick note, we also included measures of individual physiology, which I don't explain here due to time constraints. We first looked at age and sex to investigate if there was any major difference between the age-sex classes. And unsurprisingly, we found some stark differences. Females in purple are more likely to mob than males in blue, as you can see in this plot. We also found that mobbing behavior peaks at around age 8, which may be the optimal age in terms of knowledge of lions gained from past interactions and physical fitness, which declines with age. Based on these clear differences in age and sex, we divided all our future modeling efforts into 3 categories: juveniles, or hyenas less than 2 years of age, adult females, and adult males. After model selection, we found these as our top models explaining variation in mobbing behavior for adult females and for adult males. You don't need to process this figure. It's included simply to let you know that the rest of the plots I show you account for all of the variables in these models. So first, we can see that high social rank increases the probability of mobbing participation. This positive correlation between social rank and mobbing is unsurprising because higher ranking hyenas can accrue the largest direct benefits from any food acquired via mobbing. High-ranking individuals are also usually in the best physical condition due to their priority of access to resources. We also found that real-time affiliative interactions via greeting ceremonies importantly facilitated individual participation in mobbing behavior in adult females. Females that had engaged in greeting behavior during the 5 minutes before a mob formed were more likely to mob than those that had not greeted. Interestingly, greeting behavior can wipe out the negative effect of rank for low-ranking females, who, when they greet beforehand, as shown in blue, were more likely to mob than low-ranking individuals who failed to greet, as shown in purple. Because higher-ranking individuals were more likely to mob overall, greetings may be especially important to low-ranking adult females with respect to reaffirming social bonds or being recruited as a mobbing partner. Hyena participation in cooperative mobbing was also promoted by long-term social relationships. Hyenas with a higher association index, or those who spent a larger proportion of their time with the other mobbing hyenas, were more likely to mob as well. Interestingly, this was true for both adult females and adult males, despite adult males having weaker social bonds within the social group. Finally, focal individuals that were maternally related to a larger proportion of mobbers were more likely to mob than those that were related to a smaller proportion of mobbers. This is true for both adult females and adult natal males. We weren't able to test this for immigrant males. So, returning to our 3 hypotheses, we find that characteristics that suggest a stronger individual, such as being female, prime-aged, and higher ranking, increase likelihood of mobbing. Second, real-time affiliative interactions via greeting ceremonies importantly facilitated individual participation in mobbing behavior, especially for low-ranking females. Finally, we found that individuals who are close social allies or kin are more likely to mob together. Moving on to question 2, who benefits from collective action? Here, we investigated which individuals benefit from participating in cooperative mobbing in an effort to evaluate the respective benefits of cheating versus cooperating. Our past research indicates that mobbing increases the probability of hyenas feeding. So here you can see that as the number of mobs formed increases, so too does the probability of any one hyena feeding or getting at least 2 kilograms of food. However, this research did not address the individual benefits of mobbing, as we do here. So we had 3 predictions in support of this hypothesis. First, we predicted that hyenas that were hungrier or that were more motivated to gain food may be more likely to participate in cooperative mobbing at sessions with food. Here, our measure is that of belly size as a proxy for how recently individuals have eaten. And we find support for this prediction as hyenas who are obese are less likely to mob than other hyenas. However, hyenas who are obese may be less likely to mob for reasons other than lack of hunger. These are photos of the same hyena, Emma. The top one shows her with a normal belly, while the bottom one depicts a very obese Emma. We could imagine that Emma might be less likely to mob in this obese state simply because she would have to waddle everywhere and thus would move slower when potentially avoiding lions. Second, we predicted that individuals may be more likely to mob when the immediate resources are limited. Here, our measure is that of carcass size at the session. And we also find support for this prediction, as hyenas are less likely to mob at extra large carcasses, such as hippos and elephants. While medium-sized carcasses, such as wildebeest, disappear within minutes, extra large carcasses last for days in the Mara, so the risk of mobbing to gain access may be unnecessary when simply waiting will yield rewards. Our final prediction of this hypothesis is that individuals who mob should be more likely to feed than individuals who don't. In short, mobbers should reap the benefit of participation in this cooperative behavior. If we look at the 5 minutes after a mob, we do find that mobbers are more likely to feed in those 5 minutes, which makes sense because the mobbers would be the ones getting closest to the food source. This is especially true for low-ranking individuals, as low rankers who mobbed, shown in blue, were more likely to feed than those who did not mob, shown in purple. However, we do not find that mobbers are more likely to feed in the session overall. Given how quickly a group of hyenas can reduce a carcass to bones, food obtained by mobbers during or immediately after mobbing could be enough to promote this risky behavior, especially for low-ranking individuals that would not ordinarily be able to access the carcass. Note, however, that this food reward is not sufficient to entice all low-ranking individuals to cooperate, as high-ranking individuals were still the most likely to mob. So when thinking about who benefits from mobbing, or who gets to feed once this lion leaves, we find that mobbers are indeed more likely to feed than individuals who do not mob. In conclusion, please recall that we chose cooperative mobbing behavior in spotted hyenas as a prime example of a collective action problem, because the benefits derived from mobbing could accrue to individuals that did not participate in the mob. Overall, we found that cooperation in hyena societies appears to depend on the shifting contexts of individual attributes and social relationships. This suggests to us that these behaviors may be a venue in which social selection favors individuals who attend to the social context of their group mates in order to safely navigate these dangerous interspecific interactions together. So I'd like to acknowledge all of the many people who contributed to this long-term data set and to this analysis, as well as my funding sources and the Kenyan organizations who graciously permit us to do our research in the Mara. And if we have time, I can take questions. Matthew can let us know how I did at sticking to the time limit.
Well, I think maybe I'll just ask you one question for now, and then people who have questions, feel free to type them in and we can get to them at the end of all the talks. But my question, I mean, this is really cool stuff. And it immediately kind of makes me think about what we know about mobbing and crowd dynamics in humans. And one of the collective action mechanisms that we know works really well in humans is fear of costs for defecting. So my question is, do you think that, maybe speculate a little bit as to whether or not you think that there are costs to defectors and how you might start to ask that question in the system?
That's a great question. In terms of thinking about cost to defectors, the most obvious one would be to investigate if there's any sort of punishment for not participating in the mob. And I think that would be really difficult to do because of sort of what you would have to show to indicate that it's punishment and that it's not, say, an aggression due to some other reason. If I were to investigate that, I'd have to think more about how to do that. I haven't thought about it because it seems really difficult to prove, yeah, but I'll let you know.
Okay, fair enough. If other people have questions for Tracy, please do put them in the Q&A and we'll circle back to them at the end. So thanks very much, Tracy. It's really cool stuff. And I think a really a powerful testament to the power of the long-term data set. So thanks very much. Okay, our next speaker today is Connie Rojas, who is an NSF graduate research fellow and PhD candidate currently working with Kay at Michigan State. Connie studies the pathways and feedback mechanisms by which animals' behavior shapes their microbial communities and how in turn those communities shape their host's phenotype. Her dissertation focuses on functional contributions of hyena microbiome to behavior, physiology, and fitness, which is a topic that we really haven't gotten much exposure to so far in this series. So I'm really glad she's here with us today, and I will turn things over to her now.
Thank you, Matthew. Just a few seconds. All right. Hello, everyone. Thank you all for being here today. We're going to be switching gears a little bit and yes, talking about the gut microbiome in wild-spotted hyenas. And what I mean by the microbiome is the microbial communities that inhabit the gastrointestinal tract of animals. So organisms function as holobionts. And what I mean by that is that we typically have a host and its genome, but these hosts are also colonized by millions of microbes that also have their own genomes and their own functions that they're performing in the body. And the interaction between these four components, between the host, its genome, the microbes, and their genomes, shapes the phenotype of the holobiont as a whole. In the mammalian gastrointestinal tract in particular, we know that the microbiome performs key roles for the host and aids in host digestion and detoxification, and also contributes to energy homeostasis. Decades of research in the gut microbiome show that gut microbes are mostly acquired through the environment and through foods that we eat, and is shaped by our lifestyle. And we see relatively little vertical transmission of the gut microbiome from mother to offspring, and this typically only occurs after birth. We also know that the gut microbiome changes across lifespan due to a variety of factors related to physiology and the environment, disease. Another feature of the gut microbiome is that its composition tends to be highly variable among individuals. So shown here is a stacked bar plot showing the relative abundance of 4 major bacterial groups, shown here in the different colors. And we see that there's a lot of variability among individuals, so the abundances fluctuate a lot. However, when we combine the genetic information of all of these bacteria and map this information into bacterial pathways, we see remarkable consistency. So in the bottom plot, now the different colors represent different bacterial gene pathways, and we see that there's not that much variability among samples. The communities at the functional level, the communities look very similar. And these two properties of the gut microbiome are thought to be functionally adaptive for the holobiont. So if you can imagine, plasticity in gut microbiome composition enables the holobiont to respond to changes in the environment, like if there's a seasonal change in food source. And functional redundancy in microbial metabolisms preserves the core functions of the gut microbiome amidst perturbations. Like if you're struck by disease and all of a sudden one bacterial group gets wiped out, but there's opportunity for another bacterial group to come in and perform the same functions in its place. However, most of this research has been conducted in humans, non-human primates, and lab animals. It has not been longitudinal and hasn't explored the influence of certain host factors like social rank. So in this study, we are addressing these gaps in knowledge and asking 2 questions. 1, which factors shape the gut microbiome among wild-spotted hyenas? And we're focusing on host matriline, on age, social rank, and sample date as a proxy for local environmental conditions. And our second question asks, is gut microbiome composition more variable than metabolic function among individuals? And for this, we have fecal samples from adult female hyenas from 4 matrilines that vary in their rank. And for each matriline, we have samples from the mother, the daughter, and the granddaughter. And for each individual hyena, we have at least 15 fecal samples that were collected across their lifespan. And when we're looking at gut microbiome composition, we're using our entire data set. But when we're looking at gut microbiome function, we are restricting our analyses to 2 mother-daughter pairs, shown here in the blue squares. And these have 8 samples per individual hyena that were collected within a short time frame. And we are only using these 32 samples because of sequencing costs. The sequencing for this type of information is much more expensive than just simply looking at composition. So sort of the first thing that we find is that the gut microbiome composition is highly variable across time. So again, here we have a static bar plot showing the abundances of major bacterial classes, shown here by the different colors, and samples are organized from oldest sample to most recent sample. And the first thing that we see is that sort of around 2007, there seems to be a change where bacilli, bacteria in the blue are completely replaced by bacteroidea in orange. And also around this time, we have the appearance of fusobacteria in the blue that wasn't prominent in the community before. Despite these changes, we do see that there's one key bacteria that is really dominating the community, and that's Clostridia. And this bacteria is known to help hosts digest protein, which makes sense because hyenas consume a lot of meat. But even if you even notice, even within Clostridia, there's a lot of cyclical variation where there's, in some samples, it constitutes a large portion of the community, and other samples, very little. So even within this major group, there's a lot of variation. So the next step would be to try to investigate what kind of factors are possibly responsible for these changes, and also what these changes in composition mean for the community as a whole. So now if we take those bacteria and their genomes and combine them and then map these onto gene pathways, this is the picture that we get. So this is showing the relative abundances of sequences to particular gene pathways in different colors. And we're seeing that the gut microbiome is enriched in pathways related to the biosynthesis of antibiotics and secondary metabolites, which is shown here in the blue color. And this is unusual, but also could reflect the high levels of microbial competition that are found in the gut. The gene pathways are also mostly belong to functions related to metabolism. And it is important to note that only 38% of microbial genes could be assigned a function. So I could only use 38% of all of my sequence data, and this is something that the field is working in. We're trying to culture more microbes and test their functional capabilities. So this is where we are at right now. I wish we knew more, but maybe in the future we will. So now when we look at a type of permutational test, we see that Hyena identity accounts for 7% of the variance in the gut microbiome, and matrilineal accounts for an additional 3%, social rank a little bit under 2%, and Hyena age and sample date account for less than 1%. And as you can see, most of the variation in the gut microbiome isn't accounted for by the host factors that we are studying in this type of model, which is maybe it's not the most appropriate to use, and we should look at other types of analyses that might be able to pick up on a more fine scale variation. And now when we compare the gut microbiome similarity between two hyenas that are from the same matriline compared to two hyenas that are from different matrilines, we don't see much of a difference. So again, we're not seeing an influence of relatedness on the gut microbiome. However, what about when we look at age, rank, and the local environment? So now I'll be showing a series of plots that look just like this one, where we are comparing microbiome similarity on the y-axis, and on the x-axis, we have a comparison between two samples. So in this case, we're comparing the difference in age between two hyenas, and correlating that against microbiome similarity. So we're not seeing much of a relationship here. Samples from hyenas that are closer in age are more similar than samples from hyenas that are very different ages. Then when we move on to looking at the difference in rank between two samples, going from low, which means that the two hyenas are virtually of the same rank, to high, where the two hyenas are opposite ranks, or on opposite sides of the spectrum, we see a weak correlation with gut microbiome similarity. However, when we look at the difference the time that has elapsed between any two samples, we do see a relationship here. So samples that were collected closer in time tend to have more similar bacterial communities than samples that were collected further apart in time. And this is again showing how local environmental conditions are strong predictors of gut microbiome structure in this species. So now, what about when we compare samples within individuals? So we're comparing samples from one hyena against each other and not comparing those to those of other hyenas. We're just restricting this to within individuals. And like, yeah, we see that the gut microbiome changes with age, and samples that that came from hyenas that were of similar, when the hyena was of a certain similar age, tend to be more similar than samples between between hyena when they were different ages. Which again, this also represents time because we're talking about the same hyena. So age is synonymous with time here. And then when we look at the difference in rank, we see a relationship there in the opposite direction, in that samples that are of different ranks tend to have more similar communities than samples that are closer in rank. But also this could also just be an artifact because hyenas mostly do not undergo dramatic changes in their social rank across their lives. So as you can see, most of these dots are concentrated here in the low rank difference area. But it'll be interesting to sort of look into more. So now for our last question, which investigates the variability in the gut microbiome composition compared to function. Again, here we have our two mother-daughter pairs. And as I've shown in previous slides, we see a lot of variation in the composition within individuals and across individuals. However, when we combine this information and instead look at function and at the abundances of particular bacterial pathways, we see a lot of consistency. The communities look the same. And this is important to note that it sort of agrees with findings that were found in humans and macaques. And other than that, these type of analyses haven't really been conducted in other systems. So it's just really nice to see that it sort of aligns with what I thought, what we predicted. So in summary, we found that the hyena gut microbiome composition and function reflects the host's high protein diet, that gut microbiome composition is predicted by the local environment, as time between samples was a significant predictor of microbiome similarity. Within individuals, gut microbiome composition changes across lifespan, likely responding to changes in host physiology in the environment. And then lastly, variability in gut microbiome composition and functional redundancy of microbiome functions are likely adaptive for the hyena holobiome. And I would like to thank all of these organizations and funding agencies that made this research possible. And I believe I have time for questions, so we could do those.
Great, thanks very much, Connie. Really cool stuff. Let me ask you a question about the composition kind of across the group. What changed in 2007 to shift that community composition? Is that environmental? Is that demographic changes? What's happening?
So I believe, was there a poisoning? I feel like Tracy know more about this. There was certain like clan visions happening and maybe a poison event. Is that true, Kay?
Yeah, there was also a drought in there, which may have had something to do with it.
That's important to know. Good question. That's the first thing that also struck out to me, like what did happen in that time? Because this is a dramatic change.
Liz, you have a question?
Yeah, I was curious about the mother-offspring relationships, and if you looked at or are planning on looking at if the gut microbiome is potentially like transferred from mother to offspring, if there's anything known about that, and if they're more similar in earlier in life compared to later in life for the offspring and mom.
That would be a fantastic project to conduct, but sampling is really difficult of the baby hyenas, and any tiny animal, baby animal species, it's really hard to collect these fecal samples. If we could get them, that would be a really awesome study. But we do know in other systems, like in mice, that there is some transfer there. And for my postdoc, that's one of the things, one of the directions I'm thinking of going, so studying how much of it is transferred from mother to offspring, and then seeing how that community changes with development. And so, good question.
Cool, thanks.
Well, thanks. Maybe we'll leave it there for now. If folks have additional questions, which I see there's at least one, we'll come back to them later. Great. Thanks very much, Connie. Okay, moving on. Our next speaker is Dr. Eli Strauss, who received his PhD from Michigan State last year and who is now a postdoctoral fellow connected with the Beacon Center for the Study of Evolution in Action and the Max Planck Institute of Animal Behavior. Eli studies, among other things, the emergence of dominance hierarchies and social structures, both in hyenas as well as in other species. I'm glad he could join us today, and I will turn things over to him now.
All right, thank you. It's really exciting to be here, part of this really awesome seminar series. So thanks for inviting me. Just get my pointer working. There we go. Okay. Yeah, a critical piece of the social evolution puzzle, I think, is to understand how group living animals experience conflicts of interest within their groups and how within group competition is regulated. Dominance hierarchies are an incredibly taxonomically widespread social structure that regulates group conflict by determining which individuals' interests take precedent over others. I'm especially interested in dominance hierarchies in the context of the societies of nepotistic animals like spotted hyenas and many primates, where variability in kinship and social relationships give rise to corresponding variability in how the interests of different individuals align. Moreover, despite the advantages of being high ranking in these systems, rank in philopatric individuals is inherited from the mothers through a behavioral learning process that Kay told you about. And this contrasts starkly with systems where individuals compete directly for preferred social status. So today I'm going to present to you 3 short vignettes demonstrating how my research uses long-term perspectives to reveal new insight into the structures of dominance and competition within these societies. And I'd like to start by telling you a story about 2 specific hyenas. The first one is Waffles. She was born to a very low-ranking female and spent her early life being dominated by other members of her social group. However, after a few years, she ascended to the top of her social hierarchy, and her offspring are now the most privileged members of their society. The second hyena, DJ, showed the opposite pattern. Born to the second highest ranking female, DJ had a nice cushy early life. However, her descendants are now amongst the lowest ranked members of her group. So this led me to ask, what produces dynamics and dominance hierarchies, especially in systems where rank is inherited? And given that all hyenas would rather be Waffles than DJ, why do some individuals go up and others go down? So to do this, we tracked the yearly ranks of all adult females in 4 study groups over nearly 30 years. And each line in this plot depicts the rank of a single female over the course of the study. And in these ranks, we'd observe 2 types of dynamics. First, we saw active rank changes, which involve a reversal of a previously held dominance relationship and can be identified in the plot as cases where the lines cross. Second, we identified passive rank changes, which involve no reversal of a previously held relationship. And here you can see these in the plot as cases where the lines are sloping downward or upward, but without crossing. So let's start first with active rank changes. Here, based on our understanding of hyena and primate societies, we expected social alliances to be very important. So we looked at coalitionary behavior as depicted in this photo. During these types of agonistic interactions, 2 or more individuals gang up together to behave aggressively towards a third. And although we couldn't look at kinship in this study because we didn't have full kinship information for all individuals involved, prior work in hyenas and in primates has found that kinship is really important in these interactions, in that kin prefer to coalition together. So here we found that active rank changes were well predicted by coalitionary behavior. Almost all coalitions enforce the dominance hierarchy. However, in the rare cases where we saw up hierarchy coalitions where 2 low-ranking individuals were behaving aggressively against a third, in 2/3 of these cases, 1 or both of these 2 individuals came to outrank the one they were aggressing against in a future point. We also found that each individual's yearly active rank change could be predicted based on the amount of coalitionary behavior they engaged in. So females that engaged in more coalitionary aggression with their top allies were more likely to ascend in the dominance hierarchy in that year. So how is it that Waffles improved her rank so dramatically? The answer is she had a lot of coalitionary support. So if we look at her ascent in the dominance hierarchy on the plotted on the bottom here, you can see that this coincides exactly with this precipitous spike in her output of coalitionary behavior. So what leads to other hyenas like DJ showing this opposite pattern? To answer this question, we'll now turn to the second type of dominance of rank changes that I discussed earlier. These are changes that occur due to demographic processes. And I can show you an example of these at the top of the figure. So if you look at the year 2000 and scroll to the top, what you can see is that the highest ranked female died in that year, or sorry, in 2001, and the second ranked female came to occupy that highest position. In societies where rank influences when and how often individuals leave or enter the hierarchy, we expect that these passive changes might accumulate over time to lead to long-term patterns in rank change. And so here's a simulation of what that might look like. I've plotted the 3 individuals and their descendants over multiple reproductive events. What you can see is that as new individuals are recruited to the group, all those that are ranked below them move one spot further down in the social hierarchy. This leads to steadily increasing differences among individuals and on average decline in rank over time. And this is the pattern that we're expecting to see in the hierarchies of the spotted hyenas. And turning to the empirical data, this is what we see. So through passive rank changes, individuals and lineages show a tendency to decline in rank over time. If you look at the sloping lines here, sometimes they're sloping downward, sometimes they're sloping upward. But if you look on average, they more often are sloping downward than upward. If you track DJ's lineage over time, although DJ's rank only sloped downward a little bit during her life, the fact is that this process occurred to her descendants, her daughters and her granddaughters as well, such that by the time we've reached many decades later, her offspring or her descendants are now rather low ranking. If we track the mean rank of adjacent matrilineals over time, we see that this process unfolds sort of reliably on a lineage scale. So as time goes on, the differences, the mean rank in adjacent matrilines becomes more and more divergent. Finally, this amplification of small differences in rank over time also applies to the consequences of those active rank changes that I was telling you about before. So if you look at the individual marked with the diamond here, at the bottom, you see that in the mid-20s, she passed 4 individuals in the dominance hierarchy. It's kind of hard to see, but just trust me, it was 4. And if we now look at where she ranks in 2014, what we can see is that she now outranks 6 individuals that would have otherwise outranked her had she not made that change. So taken together, this long-term perspective shows how these social hierarchies unfold over large time scales as a combination of both competition via social alliances and the gradual accumulation of demographic processes. So now I'd like to turn from patterns of dominance and competition that unfold over generations to something that happens a lot more quickly. If hyenas compete for dominant status via social alliances, it's expected theoretically that infanticide by females may evolve in this system as a mechanism of competition among family groups, where females may reduce the potential allies of competitors by killing their offspring. However, very little is known about when and how frequently infanticide occurs in this system. This is partly because infanticide and juvenile mortality more generally is highly ephemeral in hyenas and in other species. In our observations, all evidence of an observed infanticide event, such as this one, was either removed from sight or consumed shortly after the event took place. So this led us to wonder if we couldn't use this long-term data set to get a better understanding of how this phenomenon works. So in this study, along with my co-authors Malit Pion and Ali Brown, and of course Kay, we examined the frequency of infanticide and the context under which it occurs. So let's start with the qualitative observations about when infanticide occurs. Of our 21 cases of infanticide, we found that it was always committed by adult females, although victims could be both male and female. And victims were typically not close kin, however, there were some exceptions to this. And killers were sometimes assisted by kin. So in this screenshot from a video, what you're seeing here is an adult female who's attacking a juvenile in her social group. And behind her are her 2 adult daughters who are in the process of chasing away the mother of that juvenile who's depicted just off screen. Finally, we found that killers were typically of high rank, whereas the mothers of the victims could be of various ranks. In particular, these last two observations are consistent with the hypothesis that these events reflect competition among matrilineals over social status. However, with 21 observations, we really didn't have enough power to conduct a robust test of this hypothesis or of any alternative hypotheses. So this is something that we'll have to come back to later once we have another few decades of data. So next we wanted to know, is infanticide a significant source of juvenile mortality? To do this, we assembled all of our cases where we knew the mortality source for juvenile mortality. And unsurprisingly, we found that these were age structured. So things like siblicide and infanticide were most likely to occur in young juveniles, whereas older juveniles were most likely to suffer predation from lions or humans. We next used these observations to fit a Bayesian multinomial model of the cause of death as a function of the age of death. And then finally, we took all of these unknown cases of juvenile mortality where we didn't know the case, and we used this model to infer how the frequency of these different known sources of mortality. And our results indicate that infanticide and predation by lions were the two leading causes of juvenile mortality in spotted hyenas. And furthermore, the probability of suffering these different mortality sources varied with age. So juveniles that died under 4.5 months were most likely killed by a conspecific, whereas juveniles that died after 4.5 months of age were most likely predated upon by lions. So overall, we found that although infanticide is rarely observable, it's actually a leading cause of juvenile mortality in the system. And we found some observations that were consistent with the hypothesis that infanticide by females in nepotistic societies arises from competition among family groups. However, some future study, I think, would add a lot more security to this conclusion. And finally, I think this suggests a potential interesting trade-off between social development and infanticide risk, where juveniles who are more social during this juvenile period and spend more time outside of the safety of the communal den may suffer a greater risk of infanticide in order to invest more time into social development. So why might it be important to invest in social development, you ask? Well, for the last section of my talk, we'll be talking about that. So you've already heard today about how important rank is in enabling hyenas to protect their interests over the interests of others. And you've heard how rank is acquired in this system via this rank inheritance process, which is a behavioral learning process. In fact, this rank inheritance process is highly reliable. So we find that 80% of hyenas acquire the exact rank that we would expect according to maternal rank inheritance. And we see here that maternal rank is highly predictive of offspring rank. However, although baby hyenas are born with a full set of teeth and the tenacity to compete with each other, they're not born with a full set of relationships with their group mates or with complete knowledge of where they belong in the dominance hierarchy. Instead, they have to learn this over the course of their development through interactions with others, and not all juveniles learn at the same rate. So in this study, Dai, Shizuka, and Kay and I investigated the fitness consequences of variability in the development of dominance rank. So here's what that variability looks like. Imagine 7 hyenas cubs living together at the communal den, born to different mothers and with different ages. And I've arrayed them on the left side here according to where we expect them to fit under maternal rank inheritance. And so if we plot their interactions over time, we see something like this, where early in life, we see many up hierarchy aggressions, so aggressions that are not consistent with the processes of maternal rank inheritance. But as development goes on, those become fewer and fewer. And towards the end of this developmental period, we see that they're almost entirely directing their aggressions according to the tenets of maternal rank inheritance. And we can plot the ranks of these same individuals over time based on the outcomes of these interactions. And what we see here is that some cubs attain or exceed their maternal rank quite early in life. So for example, cubs 5 and 6, you see here, are performing at or above their maternal rank very early on, whereas cubs such as number 3 and 4 really struggle to attain their maternal rank even towards the end of this developmental period. So in this study, we want to know whether variability in this rank acquisition process was associated with any downstream fitness consequences. And to ask this, we measured for each cub how different its rank was at the end of this communal den period from the rank that we expected it to hold under maternal rank inheritance. And what we found was that this was in fact associated with long-term fitness consequences. Cubs who underperformed their expected rank showed reduced survival and reduced lifetime reproductive success, even after accounting for their maternal rank. So to help you digest these plots, the solid lines indicate cubs born to high-ranking mothers, and the lighter color on the plot indicates cubs who were underperforming their maternal rank. And so what we see is that the cubs with the highest fitness were the ones born to high-ranking mothers and who achieved their maternal status by the end of the developmental period. And the ones who had the lowest fitness were those born to low-ranking mothers and who were underperforming their expected rank at the end of this communal den period. And interestingly and importantly, although these fitness consequences appear to be, unfold over a large time scale, the variation that's predicting these consequences is actually highly transient. So I showed you this plot earlier showing how predictable rank acquisition is in this species. And what you can see here is that that even those who are underperforming their maternal rank at the end of this communal den period are still acquiring an adult rank as expected under maternal rank inheritance. So in conclusion, I've shown you how taking this long-term approach can reveal new insights into competition within nepotistic societies. In vignette 1, I talked to you about how long-term patterns in the dynamics of dominance result from demographic processes and from competition via social alliances. And I showed you how infanticide in vignette 2, how infanticide committed by female group mates is a significant source of juvenile mortality and may relate to this competition among social alliances. And then in vignette 3, I showed you how variability in a juvenile's ability to compete against their peers is associated with long-term fitness consequences down the road. And so with that, I would like to acknowledge everyone who's worked on this, the Model Hyena project, and also my co-authors, funding institutions, and our Kenyan partners. Thanks.
Great. Thanks very much, Eli. So I think we have time for just one question. I'll take David Richardson's and kind of add on to it a little bit. I think what you told us about active rank changes is that they're driven primarily by coalitionary support. Is that right? And so what's the constraint on that? Why doesn't everyone do that? It seems like, is it just really hard to coordinate? I'd be interested to hear what you think about that.
Yeah, so I think that a lot of dominance hierarchies, and especially, and I think, definitely these coalitionary based dominance hierarchies as well, have a lot of self-reinforcing dynamics that sort of inhibit changes in the hierarchy. And I think it's actually fairly evident in the Hyena system, because what prior work has shown is that the individuals that hyenas like the coalition with the most are the ones that are ranked directly above them in the social hierarchy. So in fact, if hyenas are if a hyena is witnessing an interaction and deciding whether it wants to jump in to get involved, it almost always joins the one that was already predicted to win that interaction. So basically these coalitions sort of by default are always supporting the status quo. And so I think it takes a lot of activation energy or some sort of social coordination in order to get individuals on board to sort of, cooperatively decide to go against the grain. And so we did look at this a little bit in that study, and I didn't have the time to talk about it, but we found that individuals who coalition more frequently together were more likely to direct those coalitions up the hierarchy. So that suggests that by sort of engaging in a lot of coalitions together and getting on the same page, this might kind of provide that activation energy.
Great. Well, thanks very much. We've got some more questions for you that we'll come back to later. Great. Thanks very much, Eli.
Oh, I better stop sharing my screen.
So now we'll turn back to Kate to close us out. I've already introduced her, so I'll just return now to the amazing Kate Holekamp. Take it away, Kate.
Cancel. Okay, sorry. Well, thanks again, Matthew. It's really great of you guys to invite us. But so I just want to end up today talking about a little bit about the mediation and evolution of female aggression and dominance in this species. And I want to define that as the ability of adult females to win against adult males in dyadic contests over resources. And As you know, spotted hyenas are odd insofar as they lack a lot of the normal mammalian sex differences that we see when we look around amongst other gregarious mammals, including our own species, where males are bigger and stronger and socially dominant and more aggressive than females. Instead, females are actually, spotted hyenas are monomorphic, and females look just like males. You can see which is which here in this X-rated photo, but females are only, somewhere around the neighborhood of 10% larger than males, and otherwise look remarkably similar, including the bizarre genitalia that they sport. This is a female spotted hyena that you're seeing here, and this is her elongated clitoris, and she actually urinates, copulates, and gives birth through this. But one of the things I want to address today is the striking sex difference that we see in aggressive behavior. This is an analysis that another student in the lab, Kevin McCormick, did recently, where he was looking at the overall lifetime average rates at which females versus males emit aggressive acts. And you can see that females do this much more frequently than males do, and the average intensity of their aggressive acts is considerably higher than what we see in males. So what I want to do is take advantage of this long-term study, because I think one of the nice things about long-term studies is that they actually allow you to address questions like this at multiple levels of analysis. So some of you may know I'm a big fan of Tinbergen's explanatory scheme, where he has multiple levels of explanation for any given phenomenon in animal behavior. And so I want to run through these very quickly for you today, where we have a couple of different types of proximal explanations. So I would argue that during the course of ontogeny, coalitionary support from mothers and other kin, as you've heard from Eli, will have a big effect on their aggression and dominance in adulthood. I'm going to suggest that at least one mechanism by which this happens is prenatal androgen exposure. At the ultimate levels of explanation, I'm going to suggest that the adaptive significance or function of female aggressiveness is to improve access to food and thus enhance lifetime reproductive success. And finally, I want to suggest that phylogenetically, we have a unique combination of intense feeding competition, which is actually the operation of selection, in combination with a constraint imposed by a slow developing specialized skull in the youngsters. So I just want to take a quick look at some of these things. Eli's already showed you a lot of the powerful effects of coalitions. This has also been explored during the course of rank acquisition by Ann Ing and also by Jen Smith later on. I'm showing you Jen Smith's lovely network model here, where she's actually looking at the density of support in coalitionary attacks on others and comparing that within non-kin, distant kin, and close kin. And what you can see is that the density of close kin support networks is 7 times what the density of non-kin is. So this really has a big effect, and it happens both in food-related and other types of conflicts. And these are the ones that Eli was talking about, where the animals are actually targeting animals up the hierarchy. In terms of a potential mechanism, we had found really intriguing correlational evidence that perhaps early androgen exposure in utero appears to affect aggressive behavior. Stephanie Delaniak originally documented this in young cubs, where she looked at the hormones that females are excreting in their feces during pregnancy, and then looking at the aggressive behavior of the cubs who were in utero during those pregnancies. And so she finds that cub aggression towards other cubs is strongly correlated with maternal fecal androgens. And so we followed the same individuals and some others out into adulthood now. And we see the same pattern emerging in terms of their adult female aggression towards other adults. But what we need is an experiment. And I think I'm going to show you the only experiment that's ever likely to be seen looking at the effects of a removal of androgens from the developmental system. This was an experiment done by Steve Glickman and his colleagues at UC Berkeley when the captive hyena colony, right before it closed. And I'm going to show you 2 trials here. These are literally the only 2 trials in existence, because then the colony ran out of money and ended. But what we see is, I'm going to show you 2 trials. The first one of which is this female has just been allowed into the enclosure, and there's 2 chunks of meat, or bones and meat, tied to the fence over there. And she's going to go in and start feeding, and then a male's going to try to come join her, and you're going to see what the normal situation is for adult male hyenas in nature. They get the crap beat out of them. And then we also see the next trial is going to be the female. Actually, her mother was treated with 2 different types of anti-androgens throughout pregnancy, and she is an entirely different organism. Here comes the male. So he's appeasing her now, and That's pretty much what you see during male-female interactions in nature. Gonna go away, make another drive-by. So now here's the anti-androgen treated female and her male. Feeding within a body length of her, amazing. So I'm afraid that's about what we've got to work with, because Steve Glickman has recently passed away, and so the colony's disbanded, so that's all we're ever going to see. But moving on to the adaptive significance or function of this female aggressiveness, we see that the animal's relative social rank in their current feeding group actually determines how often they're seen feeding. So we do feeding scans every few minutes while animals are at a kill, and you can see that if your relative rank is high, even if your absolute rank is relatively low, this high rank allows you to feed much more often than lower ranking individuals. And Kevin McCormick has recently done a nice analysis where he looks at lifetime reproductive success in females on this axis as a function of their average lifetime aggression rate, and their fitness is most definitely increasing with their aggression rates. And then finally, I want to just spend a few minutes talking about the possible phylogenetic explanations for female aggression and dominance. And what I'm going to suggest is that this was shaped by two different evolutionary forces. The first one is sort of the obvious one. Well, there's selection on mothers imposed by this intense feeding competition. And I'm going to suggest that selection alone can't explain what we see in hyenas. So I invoke the constraint that is imposed by slow development in cubs of a skull adapted for durophagy, which is the ability to consume hard food. And I'm going to show you that this reduces the cub's ability to ingest even soft foods quickly during competitive feeding. So they're severely handicapped, and that handicap persists even quite a while after they are weaned, and in fact, even after puberty. So as I've mentioned, once a kill is made, feeding competition is very intense. And so yes, there's undoubtedly selection favoring female aggressiveness just under these circumstances. But if that's all there was to it, you would expect to see female dominance in some other organisms that feed competitively like hyenas do, but we just don't see that. And I think that's because they don't have, those other species don't have this unique combination of constraint and competition. So we find that in spotted hyenas, the mean weaning age is 13.5 months, but based on your social rank, weaning age ranges from 8 months to a singleton high-ranking kid to 24 months to a twin low-ranking litter. But fact is that even after weaning, cubs are severely handicapped during feeding. And the reason for that is because it takes their skulls a long, long time to develop. Here I'm showing you a sequence of known age skulls that we collected in the Mara. And you can see that this 3-month-old, its skull is just a little fragile, round chicken egg type shape. But look at this guy. This is very close to the average age at which hyenas are weaned. And you can see his skull is still very far from developed compared to, say, this older adult over here. And this is another beauty of long-term studies that allows you to integrate morphological data with behavioral data and so forth, and address a whole new suite of questions. Now look at this 24-month-old. This guy had viable sperm in his testes when he died, but his feeding apparatus is still not fully mature. So you can see his sagittal crest is far less well developed than that guy's. His zygomatic arches, cheekbones here, are very flimsy, and he totally lacks these processes on his forehead to which various muscles attach. And so I can guarantee you that if this 24-month-old were feeding with this 11-year-old, the 11-year-old would win because he can tear off and consume food much more rapidly than an animal with a poorly developed skull. So just thinking about spotted hyenas in a comparative context, there are only 4 extant species in the family Hyenae. The aardwolf is a diminutive little hyena that has lost most of its teeth, and it feeds mainly on exclusively on termites. Then we have 3 bone-cracking hyenas that have morphology specialized for breaking open large bones: the striped hyenas, brown hyena, and spotted hyena. And these 2, as the artist is showing here, rely mainly on carrion in their diets, although they will make opportunistic grabs at prey, as the artist is showing you here. These live in little tiny social groups compared to spotted hyenas, which are the only hyena that has a postcranial anatomy adapted for cursorial hunting of vertebrate prey. So here's how the animals are related to one another. And you can actually see that the striped and brown hyena are sister taxa that last shared a common ancestor with the spotted hyena here, roughly 8 or 9 million years ago. And then they last shared a common ancestor with the aardwolf here, roughly 10 or 11 million years ago. But of all these species, only spotted hyenas have large dominant females. If we look at the entire phylogeny of the Hyaenidae, based on work by Lars Verdelin and colleagues, we once had as many as 28 species of hyenas roaming the surface of the earth at once. It must have been amazing. But the original hyenas, the first ones to appear in the fossil record, are these ones in blue here, which were little small mongoose or civet-like forms that were solitary and arboreal. Then there are a large number of dog-like hyenas. These were jackal-like, and these are more coyote or wolf-like animals. And you can see that Protellus here, which is the genus in which the aardwolf is the only survivor of this entire once huge clade of dog-like hyenas that occupied all the canid niches in Europe, Asia, and Africa until the actual canids made it over there. And then we have the bone cracking forms. And the ones in orange here, Wordlin calls the transitional bone crackers. So their morphology isn't quite as well developed as these ones in pink, which are called the advanced bone cracking forms. And look at this. The first advanced one to appear in the fossil record is Adcrocuta here. And then here's the genus of the striped hyena, brown hyena, and spotted hyena. So look at this. This is the skull of Adcrocuta eczema, which is the very first advanced bone cracking form. The skull was dated about 12 million years ago. And I think you can see right off the bat that it looks remarkably like the skulls of these extant bone cracking forms. All of them are very similar. And the reason for that is because if you want to break open bones, you need to have a very, very robust skull. So hyenas can put a big bone, even the size of this one and larger, in between these two premolar teeth. And because they've got all these huge bony protuberances to which their their jaw closing muscles attached, they actually can exert pretty tremendous bite forces. So here, as you know, the two main jaw closing muscles in any mammal are these two, the temporalis and the masseter, which hangs off the zygomatic arch here. But with these massive muscles and this robust skull morphology, these animals can really exert a tremendous amount of force without breaking their skulls. So you can actually see this work Jamie Tanner did, where the reds and yellows are actually the parts of the skull that are most stressed during bone cracking. So the bone is put right here between this premolar tooth and the next one. And you can see that pink arc dissipates the stresses imposed by bone cracking and dissipates them towards the rear of the skull with these cool blue colors. And so this is a design that is apparent in every single bone cracking mammalian form that has occurred in the fossil record. The Percrocutids were a group that occurred even well before the hyenas did, and the Borophagian canids also broke open large bones, and you can see that they all have this large vaulted cranium and these monstrous, robust bone structures. So I was interested to know how much force a hyena could actually exert, breaking open a large bone. And so I was on sabbatical in Berkeley with the colony there, and I went to the local butcher and I bought a bunch of cow femurs, and I fed those to the captive hyenas. And on average, it took a captive hyena about 20 minutes to break open and polish off one of these cow fingers, which is a pretty hefty bone. But I wanted to know how much force it took to do that. So I took a couple of these cow femurs and brought them back to MSU and put them in an Instron device. And there, our engineering colleagues tell us that it requires 14,700 newtons just to break open a cow femur, for crying out loud. And we've seen hyenas break open enormous bones compared to a cow femur. So clearly they've got a lot of power in the skull to do this. But what seems to have happened during hyena evolution is that there has been a trade-off. If you want a more robust adult skull in order to achieve great bite forces, the price you may have to pay is a long time during development for skull maturation. So we know that whereas adult spotted hyenas can break open even large bones, cubs can't even eat soft foods quickly. And so they are severely handicapped. And we know this, we did performance tests, not only with goat femurs here, which we bought at a local butchery in Kenya and fed them to wild hyenas, but we also gave them 30-gram dog biscuits. And look at this, even eating something simple like compressed cereal, hell, I can eat a dog biscuit. And, it takes these hyenas their feeding doesn't reach adult levels. Their feeding speed with a dog biscuit doesn't reach adult levels until they're 34 months of age. And with a goat femur, it doesn't reach adult levels until they're 36 months of age. So this is a pretty long time to be handicapped. And what we see is that the patterns of development in all the bone cracking forms are pretty similar insofar as they all wean their cubs after roughly a year, which is very, very, very late for their body mass compared to any other carnivore, including the aardwolf, who is right here. What we wanted to do to test predictions of this this trade-off hypothesis is to inquire about whether weaning was a particularly challenging life history transition for spotted hyena youngsters. And so we actually thought this transition from the communal den to walking around in the world might be dangerous because young hyenas like this one didn't know where the dangerous entities were, like lions. And given that females are producing through that tube-like clitoris, we might expect, and it's certainly been suggested in literature, that there might be a pulse of mortality associated with reproduction. But the real challenge, if the developing skull is constraining these animals, ought to occur at weaning. And in fact, that's exactly what we see. This is a really nice analysis that Heather Watts did, where she actually started a clock on the day that cubs had left the communal den. Here she starts a clock on the day cubs are weaned, and here we start the clock at 24 months. And you see that there's no pulse of mortality at all associated with first breeding. There is a small pulse of mortality associated with leaving the communal den and going out in the world, but a really big pulse of mortality, like it goes up threefold the mortality rate as right after weaning. And we know that the mother's presence and her rank both influence cub survival after weaning. So here's the proportion of cubs surviving. These are cubs of high-ranking individuals here in the circles, and the triangles are cubs of low-ranking females. But even more impressive is this: if your mother dies here within the first few months of weaning, you are toast. Your survivorship is really pathetic compared to those whose mothers remain alive. So your mother really plays a critical role in helping you out here. And it's because of females' ability to aggressively displace others from food that high-ranking females can actually displace others, and they can wean their cubs when the cub skull looks more like this than older ones. So the low-ranking females don't wean their cubs until they're somewhere in between 14 and 22 months. But high social rank allows moms to get access to food for their youngsters. So the support for the role of constraint in addition to selection in this phylogenetic analysis is that skulls develop very slowly. Hyenas are clearly handicapped when they're feeding until they're 36 months of age. Weaning is clearly a challenging time for juvenile hyenas, and maternal presence enhances juvenile survival after weaning. So I would suggest that we see female aggression and dominance being not only unique to hyenas, but unique to all mammalian carnivores, because no other mammalian carnivore experiences this combination of intense feeding competition with slow skull development. And so we come back to these four explanations at Tinbergen's level. And I keep these separated because I think people tend to think that these coalitions and prenatal androgen exposure are somehow mutually exclusive. I don't think they are, any more than the neural substrates of birdsong are mutually exclusive with early experience of birds learning to sing. So anyway, I think it's important to think about it this way, but I will stop there and say thank you to all the agencies that have funded us and fabulous grad students that you've seen for yourself. Thanks a lot.
Great, thanks very much, Kate. Well, across these four talks, I have a whole lot to think about for the next, week of my life. But we got a bunch of questions. I think that while folks put in questions for Kay, maybe let's start back, kind of circle back in the same order that we started, and we'll end with questions for Kay. So circling back to Tracy, let me combine a question from David Richardson and also from Liz, who's had to depart us. So the question is, do individuals use ritualized greetings to recruit extra mobbers? So maybe you could tell us more about what that affiliative behavior looks like. And specifically, you talked about how that behavior is more effective in recruiting lower ranking individuals. Do you see higher ranking individuals specifically seem to target lower ranking individuals with that behavior?
Good questions. In response to the first part, I haven't tested if hyenas use greetings to systematically recruit extra mobbers. However, past work in the lab by Jen Smith has shown that Coalitions, which are, as Eli said, multiple individuals ganging up on a third conspecific, are more likely to occur after hyenas have greeted. And so if we suspect that coalitionary aggression against a third hyena is potentially similar to a coalitionary aggression against a lion, then perhaps we would expect that hyenas are using greetings to recruit mobbers, but I have not tested this. In terms of rank, I don't know the answer to that off the top of my head. I could check, or you could check in Jen Smith's 2011 paper.
A question for you, Connie, also from David Richardson. He's wondering if you can test whether immigrants coming into the group have an impact on the microbiome, if there's some kind of transfer from bringing in outside microbes potentially.
Yeah, so there's a few challenges there. Number one is that it's a continual sort of like immigration of immigrant males. There isn't like a point in time or like a point in the year where like we have an influx of 20 of them. So it's really hard to sort of test like all of these events happening right all the time. And then number two, it's really hard to get samples from immigrant males either before they were part of the clan, like in their other clan, and then also after, once they've emigrated into the clan that we are studying to get. So it's just like we're really limited by the fecal samples. But we do know that in baboons, the gut microbiome of the immigrant does change and it comes to resemble that of their new group once they immigrate. But it would be right. It is an interesting thing to note that these immigrants could be bringing in other microbes from other clans. So it's we don't have enough like immigrant samples to include, but that is definitely something that we could look into more. Great question.
Thanks, Connie. For Eli, Susan Alberts and Emily Levy were both, I think, surprised and impressed by the fidelity of maternal rank inheritance. Certainly compared to baboons, it's way messier, right? And so Susan says, I might have missed this, but to what extent is this fidelity caused by body size differences during development and as an adult?
Yeah, that's a great question. So I have some, and I think everyone has some sad left behind field work from their PhD, and my version of that is some data where I was looking in more detail at this rank acquisition process and to what extent variables like body size or maternal attendance or other sort of behavioral traits might predict the ability to acquire rank as expected. In the juveniles, I don't think the body size is going to be particularly important because so in that example I showed you with the 7 cubs arrayed from top to bottom, actually the cub number 7 there was by far the largest and oldest of that cohort of juveniles, and so it really doesn't seem to. Although it could have a slight effect early, like very early on when the cubs are really young. I don't think it has a particularly lasting effect, but that's sort of one of the things that I would like to look into. Regarding body size in adults, adult females are not especially, adult high-ranking females are not especially large compared to adult low-ranking females. So, I have to think more about that, but I don't think that is having a huge effect either.
And one other for you, Eli, and I hope that I'm interpreting this question correctly. It seems like, given the maternal inheritance of rank, it seems like there's a lot of benefits for young females to try to make these active jumps in rank, these active rank changes. And so the question is there evidence for a risky young female strategy in hyenas comparable to the risky young male hypothesis in human teenage males, where there's a real big benefit to taking on potentially risky behavior early in life because of the later life payoff?
That's an interesting question. There's a lot. It's a good question. There's a lot that we really don't know about those rank changes. I think if you're asking in the context of, for example, trying to improve in rank, presumably there are risks associated with this that we haven't been able to measure yet, but one could imagine that attempting to dominate a hyena that thinks it really should be dominating you and failing could be pretty nasty. And sort of anecdotally, we do see, you know, injuries associated with these events, and sometimes mortality. So yeah, it definitely seems like there's risk involved, although it's kind of hard for us to measure that risk. So yeah, that's an interesting thing to think about, whether that risk might correspond also to age structure and when these active rank changes take place. One thing that's very tricky about the active rank changes that I haven't found out a way to solve yet is that when two hyenas change place in the dominance hierarchy, it's very difficult to determine whether one hyena went down or whether the other one went up, if it makes any sense. You know, they both, one went down and the other one went up, but the process could be driven either primarily by one individual or primarily by another, or perhaps a little bit by both. And so, so then that kind of makes it difficult to say if a young hyena overtakes an older one, was this a ascension by a young female, or was this dropping down by an older female? But I think that you're onto something interesting there.
Okay, I want to give you the last word. I have a question for you, which is kind of a broad question. So you started out your talk or your introduction by telling us that hyenas look like old world monkeys more than they look like carnivores. So what ecological features do you think led to that convergence with old world monkeys and divergence from carnivores? And you know, do you think they're either the same or different from the same kind of constraints and benefits that you told us about in terms of female aggression?
Well, I think a lot of old world monkeys actually live in very cohesive groups to avoid predation pressure. And they don't, from what I've been able to see, engage in the same very intensive competition for resources that comes about when you have a big pile of steak there that's going to go away in 15 minutes. So in that sense, it's an entirely different scenario. But, you know, if you actually consider the fact that these hyenas apparently came to be gregarious at a moment in evolutionary history where the savannas were opening up, the forests were receding, ungulates were starting to live in these big open areas in groups. And the fact is, you know, you've probably seen this for yourself, when you're out in one of these big expanses of grass, or even, you know, shrubland in many cases, you know, you can see a kill from a long way off. And if you're a lion or a neighboring hyena clan, if you've got buddies, an individual hyena would, I think, find it extremely difficult or impossible to try and defend their food against that. And so I think the big incentive for forming a group via letting your offspring and their offspring, you know, stay in the group with you and actually needing them to protect your resources. It seems to me that all has come about, you know, roughly at about the same time in evolutionary history as lions started forming groups. So I don't think it's unique to hyenas.
Thanks very much. Thanks to you all. We went from, you know, physiology, biomechanics, dyadic behavior, collective behavior, microbiology, phylogenetics, all in one series of talks. So this has been really fun for me. Thank you all for participating. And
Well, having us, Matthew.
Yeah,
Sure
thank you for the invitation.
And for everyone else out there, next week, we'll be in the same time, same place. We'll hear from Gil McIver and Beth Archie, and go fill out the survey. So thanks you all very much. Take care.
Take care.
Thank you.